Yeah, so today is the fourth lecture, and what we're going to talk about are the very basics of neuroanatomy, and also we'll start with neurodevelopment, and we'll continue with neurodevelopment in the next class a little bit tomorrow, and then we'll move into sensory perceptions and other things, which we'll get into a little more depth, and also the experiments that are done today in lab. So, so the basic structure of nervous system, or the central nervous system, or the brain, where you have pretty much, inside a skull, a brain structure, and then you have that just goes down here, which is not shown, is the spinal cord, and which is basically the whole entire thing is protected by the skull in the head and the vertebrae in the spinal cord. And as you can see, the brains of different organisms, so these are kind of relative size of each organism, and these are the structures. So you can see that they can look quite different, but at the same time, if you cut them open and look at, if you section them and if you look at the detailed structures, the very basic plan is pretty much conserved. So which is like, if you see this front part is called a cortex, and the cortex is folded, and in dolphin or humans or chimpanzees, which is smooth in a rat or a rabbit, but it does have a cortex. It has a structure called cerebellum, which we talked for a bit before. There's a brainstem underneath it. So so the point is that they look very different, maybe superficially, but they have a lot of similarities. And of course, at the cellular and molecular level, they have way more similarities, because genetically there are a lot of conserved genes, and even the genes have similar sequences between these organisms. They have very similar molecules, and they have very similar kind of concepts or fundamentals that guide their development and function. So what is the basic anatomical plan for the nervous system? So, I mean, to understand that, you have to kind of, how do you call the dorsal ventral or anterior posterior of an animal? So here you'll see a lot of examples for mouse, because like I run a mouse lab, which we do all our experiments in mice or rats, but it's applicable to humans or monkeys too. So this is the back of the mouse, which will be our back too, which is the dorsal side, and the ventral towards the belly. And then you have the anterior and posterior. So anterior is towards the nose, and the posterior is towards the tail. So that's how we kind of, when you say, so anterior rostral, posterior caudal, so you're moving rostrally means you're moving this direction, moving caudally means you're moving this direction, moving dorsally means you are going, looking up, like if you consider this the upwards of the brain towards the ear, and towards the belly is the ventral. So, I mean, this, when you talk about neuroanatomy, we'll talk about this a lot, like, a region is more ventrally located or dorsally located or rostrally located or caudally located. So this is kind of very important. It's like if you want to, anatomy is, if you can imagine, it's just like reading a map, and map, you need to understand where the north, south, east, west are. So, and if you now, as you will see that the brains, we cannot study it as a whole. We have to take it apart, and we have to take it apart now and find the components of it and study what they do. But what we also have to do is kind of in a reproducible way. So, if I cut and you cut, I have to tell you, okay, this is the fifth. How do you describe that, and how do you kind of maintain kind of a congruent like nomenclature? So what you do is you can, so that's why we also, how do you section it, look at it when you cut it open. So you can cut it coronally, that is like this, if you're cutting the nervous system, kind of cutting a blade, driving a blade from the top to bottom, sorry, or you can cut it sagittally, which is sideways like this, or horizontally like this. So you can make sections, say a brain is few centimeters long, let's say a mouse brain, and you can take like 100 micron or 200 micron sections of coronal sections or horizontal section or sagittal sections, and then you can stain them like what we talked about, Cajal staining, and then we can assemble it and you can look at a whole brain. So that's basically what neuroanatomists do. They make sections and they kind of label whatever they want. They look under a microscope. They have the images. They now they know, okay, this part of the brain looks like this, and this cells exist here, or this molecules exist here. Now, what are the basic anatomical plans? So, if you look, as I said, like you need to know the sagittal, dorsal, ventral. So, this is the top of the brain. This is the dorsal, which I call the top of the brain, and this is the ventral part. So if you look at and this blue part is called the cerebrum or the cerebral cortex, where most of our kind of conscious perception happens. Or in a very easy term, where most of our intelligence lies. And then you have the right, and so the brain is also generally divided into right and left. So you have right cerebral hemisphere, right cortical hemisphere, left cortical hemisphere. You have the structure called cerebellum. This is a evolutionary conserved structure, which is very important for motor function. And then you have a brainstem, which has basically all the nerve branches that are connecting to the brain to the spinal cord. Because as you can imagine, we'll come to that later, that most of our information are fed from our periphery, from the skin, from the muscles and everything, to the gut and insides of our body to the spinal cord. The information that comes here, it gets processed, and the brain tells the spinal cord what to do, and basically the spinal cord executes the behavior. So that's how most of the things work. You have got the cortex, you've got the brainstem, which also have very critical functions such as controlling breathing, swallowing. These are very fundamental for survival, and so that's why if you have say a glioma or a cancer or a tumor in the brainstem, or you have a injury, it can kind of stop very basic physiological functions. Which may not happen if you, say, injure the cortex. And also, this is sagittal view. So this is, you're just rotating this, and then you can cut it sagittally, and you can look at the structure. So this cerebellum, what it looks like. And then you have the brainstem with the midbrain, and you have the forebrain or the cerebral cortex, and the spinal cord goes here. And then if you just read the textbook a little bit, you'll just gain a little bit understanding of what the spinal cord does. And what you need to really know is that spinal cord is fundamental for collecting information from most of our body, except for eyes and ears, like vision and hearing. And then it sends this information right up to the brain. And then, as I said, the brain processes the information and sends the output to the spinal cord, the motor neurons, and it tells the muscles what to do. And so this is a cross section. So if you look like this is a cross section. This is the dorsal side. This is the ventral side. So the ventral side is basically, as I said, it's facing the belly. Dorsal side facing the top of your body or your back. And then there's a cross section. So if you see, so they're in this neuron called sensory neurons, which are sitting in this ganglia. Ganglia, nothing but a bundle of neurons, a bunch of neurons sitting outside the vertebrae it's inside a bony structure, and the cell bodies here will send their axons this way, and then the axons also this way, and they don't have dendrites, and they are innervating the top of the dorsal side of the spinal cord. So the information coming this way through this red structure, the cell body, it's again sent this way, and then it can either directly talk to the motor neurons, which is a spinal reflex, or the reflexive action, as we spoke about before, and it can execute a behavior, or it can send this information to the brain to get processed and come back. So it depends on the complicacy of the behavior or how sophisticated the behavior is. The cells here can make the decision, okay, like we need to talk to the brain and ask the brain what to do, or we can decide by ourselves. So in certain cases, their decision is enough, and in some cases it's not, and they send it up and kind of get feedback from the brain. And this is a kind of fascinating structure, that the internal nervous system below the skull is covered by these membranes called the meninges. And the meninges can be like kind of simply divided into the upper fibrous dura mater. There's a space called subdural space, which has fluid in it. Then you have the arachnoid membrane, you have the subarachnoid space, and then there's a pia mater, and then you have the brain structure. So this structure carries most of the blood vessels. So the blood that needs to go inside the nervous system goes through here, and also this has innervation. So, and this is a very important structure because if you have injury to this, the brain is basically exposed, and you might have inflammation, and that can be a fatal injury. And also something like migraine happens because what is happening, or headache, if you talk about headache. What is happening in this space? So the sensory neuron, so when you're feeling, you say your head is hurting, you're having migraines, the brain itself doesn't have any sensory neurons. What do you mean by that is, what is happening inside the brain, somebody goes and just pokes your brain, you don't feel anything because there is no sensory neurons, there is no sensation inside the brain itself, but the sensation lies here. So when you're giving a headache or a migraine, what is happening is the physiological disturbances that's happening in the space, is what reflected as headache or pain in the head. So even though you feel like your whole brain or head is hurting, it's basically what's happening here, and not above and not below. So it's a kind of very kind of simple structure, yet we don't really understand this, and there's lots going on here. And then you have the ventricular system. So the ventricular system is this kind of canals or kind of fluid-filled cavities, if you may say, this kind of sitting inside the central nervous system, and it's collecting fluid from the blood vessels and or nutrition and whatever nutrients or the actual the actual components that the nervous system needs to function. It circulates all through the nervous system or the central nervous system, including the entire brain, and then it also collects the waste product and then gives it back to the blood vessels, and then it takes back to the say the kidney, and then it gets excreted out. So, and the choroid plexus, which is manufacturing the CSF, and these are the ventricles. So if you're looking from top, so this is from the dorsal side, and you make a section, kind of horizontal sections, and there is the space between as I talked about in a minute, there's a subarachnoid space, and and this is very important because in diseases like hydrocephalus, where you have kind of brain swelling, what happens is the channels that is circulating the CSF or from the ventricles, they gets blocked, and basically there's a fluid accumulation, and that causes hydrocephalus. Sorry, one second. Yeah. And then, so of course, there are various techniques now, various ways we are studying this. I'll just show you few of the most modern examples of how we can study neuroanatomy, and we can understand the different components of brain or the brain structures as they are, and what are the techniques we use, and they're not pretty commonly used, and even though they're developed pretty recently. So there's this technique called brain clearing. So what happens is that the brains that we study, mouse and rats and hamsters or guinea pigs, but these brains are not transparent. So what you generally do is make very thin sections, then you study them, then we kind of map it to like existing map, and then we say, okay, we are studying this. So the problem is that we are not, in that manner, we are not really looking at the whole brain. We're at a time we're looking at 50 micron section. And if you go by section, it's very time consuming, it's very expensive, and we don't gain a full understanding of what's going on in the nervous system. So last 4 or 5 or 6 years, that a lot of effort have been gone into trying to make the tissue clear, so that you don't have to section anymore. You can label, say, a sort of neurons and their axons and dendrites are in the kind of context of the whole brain, you can look at the intervasculature. You can look at all the vessels, blood vessels in the brain, and say a brain with a stroke versus a normal brain, and you can ask, okay, what is it so different? You can ask in Alzheimer's what happened to neurons, exactly what neurons. So it's a really powerful technique, and now people have done in humans, so they have cleared a whole entire human fetus using these techniques. So I'll just show you the video how it looks like. So it's basically going from the dorsal to ventral, and they have cleared the whole brain, and they have labeled a large number of neurons, and this shows you, in a kind of context of a whole brain, how so they're basically now zooming in and going through different regions, and how using this technique, you can go from whole brain to a single brain, just like a section, just using the same tissue. And this, the advantage of being is that these tissues last forever. You can, image it really fast. So if you section and image brains, it will take you days and if not months. Now you're able to do this in a few hours. And as I said, now people have cleared the whole brain. It's basically a whole mouse. So the whole mouse is now transparent, and you can see what the neurons are going, what the blood vessels are. So you can ask a lot of questions that you basically couldn't ask before, and you can just make observations that you just couldn't make because of technical limitations. So there are obviously other ways to look at it. So now this is a dead mouse. So dead mouse, you can't see what the neurons are firing. So kind of the whole thing about neuroscience is that neurons fire, and as a result of neurons firing, we do certain things. So my talking neurons are firing now when I'm talking. So how do you kind of assign now billions of neurons? So how do you assign? First question is, how do you assign a neuron to its function? Unless if you can't see or you can't tell what neurons are firing at that moment when the mouse is doing whatever or the human is doing whatever. The second question is, how do you understand the language that the neurons are using to communicate with each other? So as I said, there is not like a talking nuclei that is just kind of making me talk, but the talking nuclei is talking to other nuclei, which things that I have read in, say, today morning or yesterday, or versus the things that I have learned over 4 or 5 years or 10 years, and kind of taking all these facts together and then kind of translating into what I'm speaking now, right? So so the whole brain is connected, and in a very kind of dynamic way, each brain has to communicate with each other. There has to be a language, and there has to be kind of the or what do you call the computation that is going on, what are the basic fundamental principles that guide this communication between each other, and also the fundamental kind of language that the brain, the neurons in the brain are using to communicate with all the effector organs. So in this case, say my larynx or my tongue, everything is the neuron. It's talking to the rest of the brain, and at the same time, it's controlling this organ so that I can convey what would I like to convey, right? So how do you do that? So one of the, there are various ways again. So one of the very recent, a recently developed technique where they basically imaging a huge number of neurons in the brain live when the mouse is, say, running or drinking water, and they can they're just like kind of recording from the whole entire cortex, and this is called the Mesoscope Imaging. And what they're doing is, so instead of doing electrophysiology, they're taking advantage of the fact that the calcium levels are increased, in which I was talking yesterday. So calcium levels are increased when there is a synaptic release at the synapse. So high calcium means most likely there's action potential. And so they use the green fluorescent protein that is basically when there's a high calcium, it gets brighter. When there's a low calcium, it gets weaker, dimmer. And seeing this change in fluorescence, they can now say, okay, these neurons are active now. But if you look at the whole brain now, then you can say, okay, this part of the brain was active when the mouse was, say, licking to, drink water. And this is what it looks like. So if you see, this is the entire cortex, and these lights are going on and off. So then they can isolate this, and they can take each of these nuclei and make traces of it. So now they kind of, from, if you see now when they zoom out of the activity, this huge number of neurons that act kind of recording at the same time, and this will kind of eventually give you patterns that you can, with it's a, when you have the data, collecting that data is a challenge here, developing the sort of microscopes and sensors. But the bigger challenge now is to process the data, which people are trying to figure out. So in these sort of experiments now, when you are trying to understand the structure and function of brain, you basically need mathematicians, statisticians, chemists, biochemists, geneticists, mouse biologists, and you name it, right? And physicists, and you need all of them to kind of solve, from the structure to function of neurons to the language they use to communicate with each other. And then another kind of very kind of fascinating technique. So now they are looking at brain from the very superficial area, but now the brain is pretty big and it has these small structures that are very important. For example, hypothalamus, which sits at the base of the brain, is very inaccessible. You can't use a mesoscope to image that because you can't penetrate that far from the top to bottom, which is like probably a centimeter or so. So you need to collect record it from some way that you insert a lens or kind of a small microscope and record from them. And while the mouse is surviving and the mouse is actually doing things. So they have developed this technique called Miniscope Imagings, where this is a head-mounted Miniscope microscope. It's a head-mounted fluorescent microscope. So this is basically they use something it's actually something very simple. So you use a cell phone camera sensor. They have a lens and they have a small light source, which is a green LED, and they have a lens that you can install inside the brain, and it collects the light. And you can basically, what you saw here, you can do similar things from the deep brain region. Now and this happened because scientists say who were electronics engineers, or they have come together with biologists and developed something like this. And of course in humans, so you can't do at cellular resolution, you can't do such invasive experiments. And what you kind of end up doing is, sorry, one second. Yeah, so what you end up doing is something called the Fmri, which is basically MRI scan, but you look at the small volumes of small changes in blood flow. And so there are now a few labs at IRC who does that at Center for Neuroscience. And what you do in this, that you make the person go through an MRI scan, and they are kind of using the technique called BOLD. They're looking at blood flow changes, and they can kind of detect very small fluctuations. And they're asking the subjects to do a task. So in this case, so they're recording from the visual cortex, and they can see the signal goes up when the subject opens their eye versus the closed eye. So close the eye means the signal goes down. They open the eye means the signal goes up. So that is that. So this is basically the structure and function kind of structural plan of the nervous system, and then we'll just move a little bit for neurodevelopment. So in neurodevelopment, this is basically how the brains and the nervous system develop in the embryonic stage when the baby is in the mother's womb. And as you know, that neurons are post-mitotic cells, and neurons don't differentiate, kind of go to cell division when in adulthood. So they go through once and that's it, and they form, and they're stable cells and structures all through their life. And also most of the structure are fun. They grow after birth, but the structures are already formed by birth. So so most of the, say, hypothalamus, thalamus, so the different structures, cerebral cortex, they're all made by birth, and they have the fundamental. So they have the number of cells. The number of cells don't really increase. What really happens is over time, over time they grow in axons and dendrites, and the astrocytes differentiate, and that helps in tissue growth. And it's also very so one is the you can understand how different organisms, the brains of different organisms have evolved, and if you study development of the various nervous system, you can see why you can try to kind of understand why the motor system is developed differently, because say in a duck you have 2 wings and 2 legs, 2 feet, and we have 4 kind of 2 hands and 2 legs. So if you see the nervous system, you will see there will be parts which are more undeveloped in those kind of animals and more developed in ours, and vice versa. So you kind of gain an evolutionary perspective of the nervous system, why they are how they are. And also there are various neurodevelopmental disorders which you can understand by studying kind of development of the nervous system. So this is a very kind of fundamental concept that what happens during, so it is called the formation of neural tube and neural crest. So if you just study embryonic development, what you know is something called, so this is again, so if you see rostral and caudal, rostral means this is towards the nose. It's a few days old embryo. And this part of development will look very similar in us, monkeys, dolphins, and mice, and pretty much all the mammals, even in birds, in fish or birds. So what it look like? This will look something like this. There's probably a few hundred thousands of cells, and this is towards the rostral, kind of towards the nose and this caudal. And what you have, you basically have various layers of cells. So the outermost layer is called the ectoderm. The inside is called mesoderm, and so you won't see it here when you're looking like this. So that's why they're showing a cross section. So they're cutting it here and kind of turning it around and facing you and showing it. So you see the outermost is ectoderm, and then you have the mesoderm, and the inside you have endoderm. So ectoderm, or the superficial, the insidemost layer, is actually what is forming the nervous system and the skin, the outermost, like the skin too. And this is called the neural plate, the inside the brain, which is basically closed and eventually forming the neural tube, which will eventually become the spinal cord and the brain. So what happens is this will look something like this. After a few days, this will start folding, and so the mesoderm, or the inside, which will become the muscles, which will form the structure called somites, and the somites will become the musculature inside the body. And this neural tube will contain all the precursors of neurons. They will kind of what do you call it? They will divide and differentiate and become neurons and glia, and then it will start forming all the structures because they will start migrating and growing in different directions. And so what are the basic structures they form? So if you go, if you go here, so this is the head, and this is the kind of the body. This will be the spinal cord, the vertebrae, and if you now go to the top, you have the prosencephalon or forebrain. So you're zooming into this now. You forget this, so this becomes spinal cord. Inside the brain, you have the prosencephalon, which is called the forebrain, mesencephalon, midbrain, and hindbrain is the bromencephalon. So forebrain is basically you got your cortex. Midbrain, you have a different structure from thalamus to hypothalamus to amygdala, etc., or structures like that. So this is more for which is forming your conscious perception of things. This is kind of functions does your vital functions, or physiological, kind of controls your fear, controls your feeding behaviors, controls your hormone secretion, your sleep, and everything else. And this is the hindbrain, where the brainstem and cerebellum lies, which is connected to the spinal cord directly, and is carrying information forward, and it's also processing some of the very intrinsic physiological functions, such as say breathing, or kind of just say chewing, and stuff like that. And also controls your body temperature, controls your what do you call as controls your body temperature, and yeah, that's it. And then, yeah, and then if you look at the telencephalon as an example, if you look at the forebrain differentiation, so the telencephalon, the two cerebral hemispheres, so and they will become eventually bigger. As I said, the neurons are kind of by birth, they're all made, and they slowly getting to kind of grow, and from these structures called cerebral hemispheres. And so this sits right at the top, and then you have the tip, you have something called olfactory bulbs, so which are the neurons that are sensing your kind of smell and is innervating the getting innervated by the olfactory neurons. And so what are the features of the forebrain? So if you just so again, if you in a developing brain, if you're making a cross section, so you have the telencephalon at the top. So what happens is so slowly during initial development, you have kind of separated forebrains, midbrains, and forebrain, midbrain, and hindbrain. But eventually what it becomes is the forebrain goes up, the midbrain goes kind of longer this way, and the hindbrain also, like this is getting longer and this is kind of radiating outside. So at the top you have telencephalon, you have the ventricles in the middle, you have the diencephalon on the midbrain, which is kind of growing inside it, and then it becomes this different structure called, say, thalamus, hypothalamus, amygdala, and everything. So what this does is these are connected to the cortex, so this is sending information to the cortex. Cortex is sending information to them. They're connected to the brainstem, and then they are sending information to the spinal cord, as well as the information is coming from spinal cord to the thalamus and then going to the brain. So these structures are basically kind of intermediating the information that's coming in and sending it to the cortex. So some of the decisions they can make by themselves, but they're always affected by so the cortex basically can override them, but for most of the functions, cortex are generally not critical to do it, but cortex controls how and when they are done. And so, and the inside, as we're talking about CSF and ventricles, so CSF flows through them, and these are all the empty spaces of the ventricles. So if you cut a brain, you will, and if you kind of flush out the fluid, the brain inside is hollow like that, and this is because of the ventricles. And also their structure is called, say, corpus callosum, which are, as I said, the brain is basically, the cortex is divided into right and left, or the brain is divided right and left hemisphere. And the hemispheres are connected by something called corpus callosum. So these are the axons that are running across the cortex. And also there is something called gray structures and white matter structures. So the gray structures are basically the cell bodies, which are concentrated here, and then white structure are the axons, which are myelinated, which run across the brain. So that's what the gray matter, white matter, like the terms come from. Yeah, now how do you study them? So we do study developing neurons, developing nervous system in the brain. And what we do is basically that we found out how the principles, how the cells, once they're made, they're migrating. So as I said, as you would know, that brain is a very complicated structure. It's billions of neurons, hundreds of nuclei. So how are these neurons going to kind of going to the predestined position and doing their function. So how are they doing that? So there's a lot of studies, a lot of labs that are focused on these questions. What do they do is they look at how cells are migrating, when cells are kind of differentiating, or when cells are reaching the final spot, and how are they reaching, and what is making them stop at their final position. So there is a lot of questions that you can ask, and so, cortex between a very accessible region, which means that is also important because it does most of our conscious, kind of handles most of the conscious perception of our senses, and does most of the information processing. And also it's very accessible in the sense it's just sits at the top of the brain. And also cortex is something that is is most developed in humans, but there's forms of cortex in different animals too. So you can study in a lab models such as a Xenopus, it's a frog, or a fish, or a mouse, and you can kind of incorporate some of this principle to be true, to stand true in humans. So so here is an example. So it's a coronal section of a mouse in embryonic day 4. So the gestation period in mice is around 21 days. So 14 days is like kind of 7-8 days before the pups are delivered, or the baby mice are developed. And this can look at the cortex. So now they're taking a cross section. This is just the cortex. If I go to the previous picture, so they're just looking here. And you can see the cells that are the neurons, that they are kind of born at they migrate from every region, and they migrate from top to the bottom, and they settle down in different layers. So the cortex is divided in different layers, and they are innervating different areas of the brain. And so one question is how this neurons kind of going into their destiny, like where they have to end up. And so there are cells, these radial glial cells, with the acid, they kind of die or metamorphose into something else, just astrocytes later. But at this, in the embryonic development, these radial glial cells kind of become the road for them, for these cortical neurons to migrate along this and kind of settle down in layers. So this is one of the kind of mechanisms. That's how cells are going or migrating to their where they should be, and radial glial cells. So there are similar mechanisms also in the brain. And using this as a model, we try to understand that how kind of a circuit is formed, or a cortical circuit is assembled, and hopefully this kind of mechanisms are conserved across other brain regions. And the other thing is, why should you study that apart from just knowing what's going on? So there's a developmental disease where, so if you had seen this brain here, that our brain have this sulci and gyri, this folds on the cortex, which the mice or rabbit, they don't have. And one of the ideas that this cortical folds allows us to pack more neurons in the cortex, and that's one of the one of the kind of ideas that's what make us more intelligent. I'm not sure if that's true or not, but that's one of the hypotheses behind the why we have these foldings. So in this disease called Ectoderm type 1, that is a neurodevelopmental disorder, since you can have from birth, it can be genetic. And so what happens is, here these neurons can migrate properly because of defects in the radial glial cells. And when that happens, that the folds are not formed, and the brain is smaller, and eventually the patients can die out of seizure or hydrocephalus or many other kind of neurological problems. But they're generally caused by defective migration of cells along radial glia in the cortex. And also, so how do you study it? And there are many ways, again, as in anything else, there are many ways to study it. So one of the techniques I just wanted to show you, not show you, but talk about, which is called inhibitory electroporation. It's like a fascinating technique, but because what scientists do, and this is also done actually in the lab in CNS, what they do is they take a pregnant mother, and around 21 days of the gestation period, so around 14 days, they cut open the belly, and the pups are in, basically it's not like humans. So mice have generally have between say 4 to 12 pups, and these are in a chain. And what they do is they inject some DNA, to say knock out a protein or label a protein, just to observe the migration of cells. Just like I showed you here, so they can label just these neurons, and then you can kind of see how these neurons are going. Can actually see that in real time or after fixation, like after killing the animals. And what they do is that they inject some DNA in it, and they electroporate it, which means that DNA is a charge, and they pass the electrical charge through it, and the neurons can then take up the DNA, and they express the genes or proteins for the genes that kind of encoded in the DNA. And how they do it is so this is like the chain-like thing, and they know what the brain is, the ventricles are, and they're using a microinjector to kind of inject a very little amount of DNA, and then they're using these two platinum electrodes to pass the charge. And so in this paper, they basically say at what angle you are electroporating. By that, you can guide the DNA to different regions of the brain and study them. So this is kind of very interesting that how you can manipulate the cells in the brain of different regions just by changing the angles how you are giving the shock. And also, so they give an example here. So this is how actually it looks like. So what you do is you infect them, transfect them with the DNA, so that the different brain regions are carrying this fluorescent protein, the GFP here, and then you can let the mouse grow. You then put the embryos back in the mouse the mom, and you sew it up, and the mouse the mother basically carries this embryos to birth, and when the mouse are born, they can even grow up for that, and then you can take out the brain and look at them, the cells that you have labeled. So depending on the time you have injected, you can see different types of cells are being labeled. You can knock a protein down, and you can understand the consequences. So say lissencephaly, the gene causing lissencephaly, you can knock out in few cortical neurons, and then you can say how those cortical neurons compared to controls, how they're differently migrating, and you can draw conclusions. Okay, this protein is really important for this day to this day migration, and if you don't have the protein, this is what happens. And probably that's what the mechanism that causes, say, what are the defects seen in humans because of lissencephaly. So this is another example of a very kind of fascinating technique that are regularly now used in labs to manipulate cells in the embryos and study neurodevelopment or cells in cortical migration. So that's that, and so question time. Yeah. So anybody?
So I think about in slide 4 or something, you had talked about the spinal cord decides what decisions to make based on how complicated it is, and then otherwise it passes it on up to the brain. Could you
Right.
like elaborate more on how exactly this is done? I'm not
Yeah.
sure whether
So
there's like a molecular level mechanism or not.
So it's a yeah. So the basic thing is, so say like for example, so there's a candle, right? And then you don't know say you're kind of walking around and there's a candle on the table, and then you by mistake you touch the candle, like touch the flame of the candle, and it's really hot, and you immediately you move your hand from it. So that's called the spinal reflex. So in the spinal reflex, it's basically so simple that the sensation can go to the spinal cord, and there is something called interneuron, and then there's a motor neuron, and the information is within the spinal cord itself. It's being processed. I think it was here. Right. Yeah. So basically, this is a sensory neuron, which is taking the information that you touch the flame, and the heat comes through the sensory neuron to the spinal cord in the dorsal side, and there's the interneuron between it, and the information goes from here to that neuron to the motor neuron, and immediately, like the motor neuron, so the sensory neuron said that innervating your fingers are connected to this. It's kind of corresponding part of the spinal cord in your vertebrae, and there is a corresponding motor neuron too, which is innervating your hand. So this is all kind of anatomically segregated, and so when this motor neuron carries information, hey, move the hand from there, you don't even have to think of it. You just do it, kind of, you can't even stop it sometimes. No, I mean, unless you're thinking about not moving it, so you can do it. But generally, your instinct will be just to move it, and you don't need the brain for that. But say something that I study, say like escape responses. So what happens is like you barefoot walk on a beach or something, not beach, like say, in a summer afternoon, there's a delivery guy came to deliver something, and you just walked out barefoot on the road. And what is going to happen is like, kind of a lot of you, you know, your whole body needs to, and then you find it too hot and you run out of that from that road. What happens is, like, you need to use your entire body and you think where to go, what will be the closest coolest spot so that my feet don't get burned. To make all those decisions, this circuit is not sufficient then, because you have to look for the place, you have to find the place, you have to make the jump, you have to decide how fast you have to go, how soon I can reach the coolest spot. So for that, all this computing, all this information, this circuit is not enough. Then it takes it to the brain and it gets processed. So that's the idea. Did that make sense?
Yeah, thank you.
Well, yeah, next.
So,
Yeah.
so when you were talking about lissencephaly, you basically said that because of improper migration of neurons, the brain does not have the convolutions it normally has. So, does
Right.
that mean that neuron migration is exclusively responsible for creating convolutions in the brain surface?
So one of the mechanisms, so that's only one of the mechanisms, because as I said, like the folds are one of the reasons they are formed like that is because to kind of house the huge number of neurons that are there. So migration is only on one of the mechanisms now. So it can be because of the supporting cells, the glial cells that actually kind of putting the neurons together. There, it can be because of not enough number of neurons are generated, so that there can be folds. It can be premature cell death, and it can be a number of reasons. So that's not the only one.
Okay. So,
Yeah. Anybody else? Yeah. Anybody else? No. Okay, so that's that, and then tomorrow we'll talk about the sensory perception and the sensory systems. So, all right, bye.
Thank you, sir.
Yeah, thank you.